The preservation of biological life depends on the accurate transmission of genetic information. In the standard flow of the central dogma, DNA serves as the template for messenger RNA, which is then translated into the primary sequence of a functional protein. This translation relies on the genetic code, a system where triplets of nucleotides, or codons, specify the addition of one of 20 amino acids to a growing polypeptide chain. Mutations occur when the underlying nucleotide sequence is altered. In a silent mutation, a single base pair is swapped, but because the genetic code is redundant, the resulting amino acid remains unchanged. The primary structure of the protein is preserved. However, a missense mutation results in a codon that specifies a different amino acid. This single substitution can alter the chemical properties of the protein, potentially disrupting its folding or its active site. This is the mechanism behind sickle cell anemia, where a missense mutation in the beta-globin gene replaces a hydrophilic glutamic acid with a hydrophobic valine, causing the hemoglobin to polymerize under low oxygen conditions. If the base substitution creates a premature stop codon, it results in a nonsense mutation. The ribosome detaches early, producing a truncated protein that is typically degraded by the cell. More catastrophic errors occur when nucleotides are inserted or deleted in numbers not divisible by 3. These are frameshift mutations, which physically displace the reading frame of the ribosome. Because the triplets are read sequentially, a single insertion scrambles every subsequent codon in the sequence. This usually leads to a completely non-functional product or an early termination. Frameshift mutations are frequently observed in severe forms of thalassemia, where the inability to produce functional globin chains leads to profound hemolytic anemia. These errors form the genetic basis for inborn errors of metabolism. When both parents carry a single non-working copy of a gene, there is a 25% chance that a child will receive 2 abnormal genes, resulting in the clinical manifestation of the disorder. Mapping these structural errors, from single base swaps to the destruction of the reading frame, explains the biochemical transition from a healthy metabolic pathway to a systemic disease state. Genetic information is not expressed at a constant rate. It is governed by regulatory networks that respond to the metabolic needs of the organism. In prokaryotic organisms, this regulation often occurs via the operon, a single transcriptional unit where multiple genes are controlled by a shared promoter and operator. The lac operon is an inducible system. By default, a repressor protein blocks RNA polymerase from transcribing genes for lactose metabolism. When present, lactose binds and releases the repressor, allowing the polymerase to transcribe the genes. Conversely, the TRP operon is active by default, continuously synthesizing tryptophan. When tryptophan levels are high, the excess amino acid activates the repressor, halting further production to prevent wasting energy. Eukaryotic cells employ layers of regulation that far exceed the complexity of bacterial operons, utilizing localized sequence elements and global modifications. Transcription factors bind to enhancer sequences and promoter regions, like the TATA box, to recruit and stabilize RNA polymerase II at the start of a gene. Regulation also occurs at the epigenetic level. Adding methyl groups to DNA typically silences genes by recruiting co-repressors, while histone acetylation opens the chromatin structure to promote active transcription. These same biological mechanisms, the specific binding and enzymatic cutting of DNA, have been adapted into the tools used in recombinant technology. Recombinant DNA technology uses these molecular principles to isolate and combine genetic material from different sources, creating custom sequences for research and therapy. The process begins with restriction endonucleases. These enzymes recognize specific palindromic DNA sequences and cleave the sugar phosphate backbone, often leaving single-stranded sticky ends that are ready to hybridize. A target gene can then be ligated into a bacterial plasmid vector. DNA ligase seals the fragments together, forming a circular recombinant molecule that can be introduced into bacteria for mass production. This technology is utilized to produce recombinant human insulin, providing a consistent supply of medication for patients with diabetes by expressing the human gene in bacterial cultures. While recombination allows for production, the polymerase chain reaction, or PCR, provides a method to amplify a specific target sequence from a complex genomic sample. In the first step, denaturation, the reaction mixture is heated to 94 degrees Celsius. This thermal energy separates the double helix into two single-stranded templates. As temperature drops, synthetic DNA primers anneal to complementary sequences on the single strands, flanking the target region. Finally, a heat-stable DNA polymerase extends the primers, adding nucleotides to complete the synthesis of the new double-stranded DNA. By cycling through these temperatures, the target sequence is doubled each time. This results in the exponential amplification of the mutated gene, making it accessible for diagnostic testing. Once a sample has been amplified or purified, clinicians use blotting techniques to identify the specific macromolecules associated with a disease. Most blotting procedures involve three core phases: separating molecules by size via electrophoresis, transferring them to a synthetic membrane, and detecting them with a labeled probe. To categorize these techniques for clinical exams, use the snowdrop mnemonic. Aligning the names of the blots on the left with their target molecules on the right creates a consistent reference. The southern blot detects DNA sequences. The northern blot identifies RNA sequences and gene expression levels. The western blot detects specific proteins using labeled antibodies. The snowdrop framework serves as the conceptual anchor for navigating laboratory diagnostic questions. Clinically, these concepts form a single diagnostic pipeline. We can trace how a root DNA mutation propagates through the transcript and ultimately produces a dysfunctional protein. This allows us to select the appropriate tool for any stage of the process: PCR for the gene, northern blots for mRNA, and western blots for protein. For rapid recall, remember the structural impact of mutations. A missense mutation is a single unit swap, while a frame shift causes a major sequence shift that destroys downstream function. For regulation, distinguish the logic of operons. Lac is induced by its substrate, while TRP is repressed by its product. And for laboratory identification, southern corresponds to DNA, and western corresponds to protein. Consider this clinical scenario. If the objective is to confirm the presence of a specific viral protein in a patient's serum, which technique is most appropriate? The correct answer is C, the western blot. Since the target is a protein, we must use an antibody-based detection method. Next, consider the regulatory logic of the Lac operon. How is the system categorized? The correct answer is B, inducible. Because the presence of the substrate, lactose, is required to turn the system on, it is an inducible regulatory circuit. Mastery of these topics requires moving past isolated definitions and understanding the mechanical flow of genetic information. By linking molecular errors to their regulatory logic and diagnostic tests, you can accurately interpret complex clinical data.